Okay, so this is where we left off in some of the classes. Some of the classes are a little bit more ahead of this, so if you're ahead of this by a little bit, just fast forward to the part where we left off in your class. But I think in 6th hour we left off with just going over this, and so we're a little ahead in some other classes. So anyway, so remember that this was we were using the liver and the lens cell and how they have the same genes, the albumin and the crystalline gene in both cells, but only in the liver cell the albumin gene is turned on, and only in the lens cell the crystalline gene is expressed. And this is because they have different activators available, and each gene needs a different combination of activators. So the right combination to bind to the control elements for the albumin gene are only in the liver cell, and the right combination of the activators that are needed to bind to the control elements for the crystalline gene are only in the lens cell. And so that's how we get differential gene expression, because different genes are turned on in the different cells, and therefore different proteins are made, which then helps with the different function of the various cells in our body. And so that's one way that we, as eukaryotic organisms in our cells, control transcription. So this is what we're controlling. We're turning these genes on based on whether or not these activators are present. So this next bullet goes with that picture. So the combination of transcription factors binding to the regulatory regions at any one time determine how much, if any, of the gene product will be produced. And so with this, you're looking at the combination of transcription factors. So you have to have the right combination. All right. So unlike the genes of a prokaryotic operon, coordinately controlled eukaryotic genes have a promoter and control elements. So this is new, the control elements to eukaryotic cells, not in prokaryotic cells. The same regulatory sequences are common to all the genes of a group, enabling recognition by the same specific transcription factors. So what does that mean? Let's review. So in a prokaryotic cell, we had the operon, right? And so we had the operon that had the promoter region, had the operator region, and then you had your different genes. All right, so it could be gene A, B, and C, let's say, like for the lac operon. And these genes are all turned on by one operator. In eukaryotic cells, each gene has a specific promoter, or said turn on by one promoter, I meant up here. So in a eukaryotic cell, if this is your DNA, and this is gene A right here, and this is gene B, and then it has their own promoter for each one of these. So each gene has their own promoter, and remember, eukaryotic cells have those control elements as well. So I'll put up here control elements, and you have to have specific activators that bind to those control elements. So what does it mean by the genes of a group? If the genes operate or have a similar function, then oftentimes the activators, the control elements that control them, the control elements may be the same. So what does that mean? So let's say, for instance, that gene A and gene B make enzyme A and enzyme B. So let's say you have molecule 1 gets converted to molecule 2, which gets converted to molecule 3. Molecule 1 to 2 is catalyzed by, let's say, enzyme A, and molecule 2 to 3 gets catalyzed by enzyme B. And so this is a pathway, this is a metabolic pathway, and in order for this to happen, and therefore to make molecule 3, you need both enzyme A and enzyme B. So it makes sense that they need these two genes that make both of these enzymes needed to be turned on at the same time. So therefore, the same combination of activators would turn on both of these genes. So they bind to that's supposed to be binding to the control elements. The same combination is needed, which means then if those activators are present, both of these genes would be turned on, and that makes sense because you need both of those genes to do your metabolic pathway. And so that's the same kind of idea as one promoter region for like the genes that control making the enzymes to digest lactose. So you need all three of these enzymes, so they're turned on all at the same time because you need all three of them to do the job that they're involved in. And so that's how this is accomplished in eukaryotic cells, and that's what these two bullets are trying to get at. All right, so that's how we can control transcription. So then we regulate after transcription, so post-transcription. So now we have our mRNA. So what happens after transcription? Remember, mRNA processing occurs. So transcription alone does not account for gene expression. So just because we've gone through transcription is making your mRNA. We don't have a protein, and therefore we definitely don't have a phenotype or an expression of that gene yet. More and more examples are being found of regulatory mechanisms that operate at various stages after transcription. So that's what the rest of this is about. So we're going to kind of go through protein synthesis and look at this. So such mechanisms allow a cell to fine-tune gene expression rapidly in response to environmental changes. And we'll talk about that here as we go along. So let's look at the first thing that happens after transcription, and that's RNA processing. So we've already kind of talked about this, treating exons and introns differently. So in alternative RNA splicing, different mRNA molecules are produced from the same primary transcript, depending on which RNA segments are treated as exons and which are treated as introns. So if we look at this here, this is your DNA. So these are your exons, which means then all of the lighter parts are your introns. So we're going to say that this is the start of the gene. So right here, that's your gene. And so I'm going to put an I here. These are all introns. So then remember that when you make your mRNA, so this is transcription. You make your mRNA, the primary RNA transcriptor, sometimes called the pre-mRNA, has both introns and exons. And so that's what we see here. So it has the exons and the introns. But remember that this is RNA processing. Both of these arrows are during RNA processing. What do? We cut out the introns, and add cap and a tail. And so here, that's what we see here. So we have the cap and the tail, but notice here, what they're trying to show you is that they have spliced these differently. So meaning that all of these introns necessarily weren't considered introns. So here, if we go in this direction here. So these were all exons here. And all the light colored, this was an exon, and this is an exon, according to this up here. And all the lighter ones are introns. Notice here what has happened. I'm going to do this one in one color and the other one in another color here. So to get this right here, it kept this, treated this as an exon, treated this as an intron, treated this as an exon, so that's why we have dark and dark, because these guys are spliced together. Treated this as an intron, cut that out, we don't see any light here, and spliced this exon with this. Treated this as an intron, but look at here. Do you see this exon here in this? No, it cut this exon out, so it treated this exon as an intron instead of as an exon. And then it treated this as intron as well. So therefore, it spliced this piece to this piece, and so these 4 pieces all make up. I'm going to put here in orange here. These guys here. All right. So that's what we see here. So that's what happened to get this RNA. To get this RNA, I'm going to do in blue here. So, treated this as an exon. That's what we have here. That's the cap and the tail here. So treated that as an exon, and it did treat this as an intron. So I'm going to put another blue X here. So it did treat that as an intron and splice the exon together, so that's why we have the black. We don't have this long part here, so it did treat this as an intron. And so these 3 exons are these 3 long spots here. But then look at it, treated this as an intron, but this is still in there, so it didn't cut that part out, it's still in there. So it did treat that as an exon. And cut out this part and treated this as an exon. So this one treated, just like the DNA, when we labeled the exon, it treated everything as an exon that we had labeled and cut out the introns, whereas this one did an alternative splicing. It treated one of the exons as an intron. And so therefore this sequence here, I'm just going to kind of color code it, is because of cutting out what I've X'd out in blue. And so that's called alternative splicing or alternative RNA processing. And so now so I'm going to put here from the same DNA, you can make different mRNA molecules depending upon what you treat as an exon and as an intron, leading to different proteins made. Oops, sorry. Different proteins. And that's how you get that. So then, going back to our, go back here, so where we're at in this whole process, let me go back to this picture here. So we've talked about making the gene available with adding chemical groups. We've talked about the role of activators and control elements in transcription. Now we've talked about mRNA processing and how we can control based what kind of mRNA is made based upon what we consider introns and exons. And then now the mRNA is going to leave, and we're going to be in this part here. So the mRNA can either go to translation or be degraded. And so that's where we're at in the big picture here. So here we have mRNA degradation. And so the lifespan of mRNA molecules in the cytoplasm is a key to determining the protein synthesis. So how long does the mRNA stay? And then if it stays longer and is not destroyed, you can make more protein from it. So the mRNA lifespan is determined in part by the sequences in the leader and trailer region, so the beginning and the end. So we have this thing called RNA interference. Now notice here it says RNA interference by single-stranded microRNAs. So RNA is single-stranded, remember, so it has the A's, the U's, so you have the sugar phosphate backbone and you have the A's, U's, C's, and G's, and so on. And so it's single-stranded. Single-stranded microRNAs. Micro means small, right? So very small little pieces of mRNAs. These are called often miRNAs. Micro mi. And what they do is help to degrade or break down RNA, mRNA, or block its translation. So going back to that big picture here, that's this part in the in here. So microRNAs help with this part here with the degradation, or if it doesn't degrade, it can stop it from being translated. Either way, if it degrades it or so it breaks it down, obviously translation isn't going to occur, or it can actually block it by binding to the mRNA. So let's look at how that takes place. So the phenomenon of inhibition of gene expression by RNA molecules is called RNA interference, or sometimes they call it RNAi. So a small little RNA molecule actually interferes with whether or not, so you've made an mRNA, it's in your cell, you should be able to make proteins, but then these little guys, these microRNAs, come into play, all right? And so how are these micro mRNAs made? So here is an RNA molecule. So the RNA is single-stranded. So that's your RNA single-stranded. And then notice that it doubles on itself and forms hydrogen bonds. So you could have an A with a U and a C with a G and so on. So they can hydrogen bond to one another. There's an enzyme that they just call a dicer molecule. You just kind of need to know the overall process here. The dicer, they call it that because it dices or cuts up the RNA into small RNAs. These small RNAs, so these are small RNA molecules. And then in the cell, they proteins bind with it, and the hydrogen bonds break, and so you have a single-stranded mRNA molecule bound to this protein complex, and so that is your microRNA or miRNA. And so this part here is the sugar phosphate backbone, and these little guys here are your bases coming out, and so you have like A, C, G, U, all right, and so they're single-stranded there. So back when we wrote that they're single-stranded microRNAs, and so that's what we have here, small RNA, single-stranded. So what happens is in your cell, let's say you've made your mRNA, so this is your mRNA molecule. So we've gone through this process here. We've gone through the chromatin changes, so we expose the DNA. We've gone through transcription. We've gone through mRNA processing. So you have your final mRNA leaving the nucleus of the cell. When that happens, sometimes the mRNA can be met with a microRNA. So that's what we see here. If it's met with a microRNA, it's going to block, and this part isn't going to happen. Translation isn't going to happen. And so why isn't translation going to happen if your mRNA that you've just made in the cell meets this microRNA? So the microRNA does two things, one of two things I should say. The first thing is that it can degrade the mRNA, so it breaks it down. So it breaks down mRNA, which equals no protein made. I'm sorry, break down mRNA equals no protein made. Or look at the second thing it does. So this is path 2. It actually blocks it. It physically blocks translation, I should say. So here's, what is this little guy, do you think? Translation, building amino acid chains. This is your ribosome. So it goes to the ribosome, tries to do translation, but remember the ribosome reads, it's read in groups of 3 and it adds the amino acids. Well, what happens is, what's going on right here is the miRNA, the microRNA, hydrogen bonds to the mRNA and physically blocks translation. So translation can occur because the ribosome get a roadblock here and it physically can't occur. So so either one of these stops translation from happening, and so that's another way to control whether or not a gene or the gene is actually going to be expressed. So sometimes we can go through this whole process of mRNA, and then these microRNAs block the rest of the process, and so therefore you still don't get your gene expression. All right, so then what's the next step here? So if we look at the overall picture here is controlling translation, actually building your polypeptide. So initiation of translation of selected mRNAs can be blocked by regulatory proteins that bind to sequences or structures of the mRNA. Alternatively, translation of all mRNAs in a cell may be regulated simultaneously. All right, so initiation stops. You can be blocked by these regulatory proteins, so they can actually bind to the mRNA and stop translation from happening as well. So let's say then that so there's no mRNA that degrades no microRNA that degrades the mRNA, and mRNA is able to be translated, and you actually get your polypeptide. The last thing here is processing and changing the protein changing the polypeptide chain after it's made. So you have a chain of amino acids, but that necessarily doesn't mean that the protein's done. So after translation, various types of protein processing, including cleavage and the addition of chemical groups, are subject to control. So cleavage means cutting off certain things. The length of time each protein functions in the cell is regulated by means of selective degradation. So selective here. Example, that's supposed to be a colon here. Example, remember, we'll look at here cyclins that regulate the cell cycle. So the cyclins will attach ubiquitin to a protein, and that means it's marked for degradation. And I'll give you an example. I'm going to show you a picture here in just a second. And there are these proteasomes that are giant protein complexes that bind protein molecules and degrade them after, I'm going to put here, after they are, quote unquote, tagged by ubiquitin. All right, so let's look at the next picture here because that shows you this process. So here's your protein to be degraded. So how does the cell know whether or not to degrade the protein? It's tagged with this molecule called ubiquitin. So ubiquitinated protein. So it means that ubiquitin has been added. So I'm going to put here in parentheses that we add ubiquitin, and that, put an equal sign, targets the protein to be destroyed by a proteasome. Okay, so therefore, here comes in, so this is the tag, hey, come destroy me. Proteasome comes in, it's the protein. The protein enters into the proteasome, and these are the protein fragments. These are peptides, small chains of amino acids. They're made out of amino acids. And so these guys can be recycled, and the proteasome and the ubiquitin can be recycled, and the ubiquitin can be used again to add to the protein, and the proteasome also does that. And so therefore it destroys it. So therefore all the protein that you've made doesn't always stay in the cell. So it may do a job for a certain period of time, and then you don't need that protein anymore. So this is how the protein is degraded. So overall gene regulation accounts for some of the phenotypic differences between organisms of similar genes. So you can have similar genes, but they're not all turned on. Even if they're turned on, some of the proteins that are made stay longer than others, and so we can destroy them, and so that's also part of your gene regulation. So I added in here to just pull in some, remember when we talked about in Chapter 11 cell communication, because we talked a little bit about gene regulation there. So I threw this in. So notice this is figure 11.2. So this is back from cell regulation. This is a review from Chapter 11. It's not cell regulation, gene regulation. So example, mating pheromones in yeast trigger mating gene expression. So the binding of factors to receptors on specific yeast cells trigger changes to happen. They turn on certain genes that lead to their fusion. So we've gone over this before, but I just wanted to relate that to this chapter as well, to kind of bridge some across the chapter. So you have mating yeast cell alpha and mating yeast type A. Alpha makes alpha factors, but the A has a binding site, a receptor for those alpha factors, and then the mating type A makes the A factor, and the alpha yeast cell has the appropriate receptors there. What happens is it triggers them to mate, and so what has happened when they bind, the binding triggers a signal transduction pathway. Transduction pathway, which leads to the activation of genes, which leads to the mating of the yeast cells. All right, and so that's a quick little review of that. And I did another one also from chapter 11. If we look here, so this is a review also. And so this is with the activation of a gene by a growth factor. So here's the cell membrane, and that's the receptor. So this is the lipid bilayer. This is the receptor, growth factor. This is the ligand. Remember that? So that's reception. Binds to the receptor, it doesn't enter it, triggers transduction, so that phosphorylation cascade, and eventually this molecule enters into the nucleus, which activates a transcription factor. So a transcription factor binds, remember, to the TATA box and triggers the turning on of a gene, but this transcription factor is normally inactive. It's not active unless this molecule here activates it. It phosphorylates it and becomes active. And so, therefore this gene would never be turned quote unquote on and make the mRNA, that's transcription, if this growth factor never bound to the cell in the first place. And so, because this would never have been phosphorylated. So that's another example pulling in from chapter 11 that we've talked about before. Okay? And then certain genes aren't turned on and off. So certain genes are continuously expressed. That is, they are always turned on. So example, ribosomal genes that make rRNA. So these don't even make proteins, so certain genes don't turn on and off. And so this gene, if this is your DNA, this gene makes RNA and never makes a protein. So remember that we also have genes that make tRNA. And then we have genes that make proteins, that code for making proteins. So then we have genes that code for making an mRNA, and then it's the mRNA that will go to the ribosome and be used to make a protein. So not all of our genes are actually making and coding for proteins. All right, and so therefore these can be continually expressed and aren't turned on and off. All right, and the last thing is looking at now looking at it from an evolution standpoint and look at how we get different genes. Like when we've talked about genetics, like big R, little r, the little r is usually a mutation of your wild type. And so let's talk about how that leads to genetic variation. So the basis of change at the genomic level is mutation, so we know that, underlying much of the genome evolution. The earliest forms of life likely had a minimal number of genes, including only those necessary for survival and reproduction. Obviously, reproduction is very important for the survival of the species. The size of genomes has increased, though, over time, over evolutionary time, with the extra genetic material providing raw material for gene diversification. So basically, we had very basic genes, all right, for survival. And over time, more and more mutations have happened and so on. And these mutations, some of them are good, some of them are bad. And sometimes when the mutations occur and they're good, it leads to a new gene in the population, and then that gene can be passed on to the next generation, to their kids, and then to their kids, and then over many years, thousands and thousands and thousands of years, you get a new gene incorporated into the population. So let's look at some ways that that could occur. And the first one is duplication of chromosome sets. So this can happen in plants. So accidents in meiosis can lead to one or more extra sets of chromosomes, a condition known as polyploidy. And I'm going to put here in plants, because remember, if we get more than one chromosome pair, if we get 3 of every type of chromosome, we don't survive. And so what does this mean, polyploidy in plants? So let's say the plant has 4 chromosome number 1s instead of 2, and 4 chromosome number 2s, and so on and so forth. So how does a plant get that? And so a plant has, so this plant would be 4N because it has 4 of every kind of chromosome. How does the plant get to be 4N? What does that mean? That the egg cell had to have 2N and the sperm cell had to have 2N. So how does the egg and a sperm cell get to be 2N to get this 4N kid? And so what has happened is that there must have been here the egg and the sperm had a mistake happen, and the chromosomes didn't separate properly. Correctly, leading to 2N gametes instead of N. All right, so how could this lead to diversification and so on? So what happens is it says the genes in one or more of the extra sets can change from one another by accumulating mutations. And these variations may persist if the organism carrying them survives and reproduces. So let me explain what that means. So let's say, so now this organism is 4N. So it's different than its parent organism that's not 4N. The parent organism was just 2N. And so let's say a mutation occurs here. So let's say, write this over here. So this is a mutation in a gene. If that mutation, so that mutation is now different than the other forms of the gene. So there's a gene on each one of these. So this one's different because it was mutated. That mutation can be good or bad. If it's either way, it can be passed on to the kids. And can become a new gene in the population. Becomes a new gene in the population. And so therefore, duplication allows for the old gene to stay intact and then a new gene to be entered into the population. So let's look at this. So the title of the next section is on this side, sorry, duplication and divergence of DNA segments. So let's look at that, of how that can happen. So another way that can happen in non-plants, because polyploidy, like I said, occurs in plants. If this occurred, this polyploidy in humans, it wouldn't work out. So how can duplication and mutations cause diversification in us, and where polyploidy doesn't happen? So it can happen when we have unequal crossing over. So unequal crossing over during prophase 1 of meiosis can result in one chromosome with a deletion and another with a duplication. So deletion, remember, is missing, and the duplication has extra of the same gene. So let's look at this picture. I'm going to do some color coding here. So these are non-sister chromatids, but they're out of a homologous pair of chromosomes. So this is a homologous pair, and they're and let's say chromosome pair number 1. All right, and they color-coded it differently. So you can see that this is the leg of the darker chromosome. This is the leg, the arm here of the lighter chromosome. And it's hard to see here, but you can see on yours that there's matching parts here of the chromosome because they're homologous. So you have a gene for whatever protein, and this one could be a big R, this could be a little r, or whatever. So this is your genes. I'm going to just kind of go over it like this so we can color code it. So the gene is in purple. And so what we're going to do is cross over. So that's what we see here, crossing over. So I'm going to color code these two parts here. This part and this part. Now, when crossing over occurs, they're supposed to, when the arms literally cross over, they're supposed to cross over in the same part. All right, so like these two would cross over at this point, or this point, or this point, and so on. So they're crossing over at the same point. So what happens here is unequal crossing over. So just look at this little picture here. You see how where the site at crossing over right here, that it's unequal, that there's more on this arm down here and then less on this arm. So let's look at how that relates from this picture to this picture. So this right here is the site of crossing over. So let's look at where the location of these genes are. So this gene is right here, and this gene is right here. And then this orange spot on the chromosome is where it's crossing over. And this blue spot on this chromosome arm is right here. So what's happening is they're crossing over, instead of crossing over at this spot here, they're crossing over here. So it's further down the arm of the darker chromosome, which is why there's less here. So what are they going to swap? They're going to swap these parts right here. So when this exchanges over here, that's going to have more DNA and more genes, and this part here less. And so that's what we see here. So I'm going to write that down. So let's write up here that instead, let me make it so you can see, instead of crossing over at sites across from each other, they cross over at different spots. On each homologous chromosome. So, all right, so that's these two spots here. So then what happens when they're done crossing over and they actually swap this part, so they're going to swap this part and this part here. So what happens is, remember that they switch DNA from the site of crossing over to the end of the chromosome. So from the site here, they're going to switch from here to here, all right? So looking at this picture, the smaller section is going to be switched with this larger section. So then let's see what happens at the end. So now let's look at the overall big picture here. This long piece of the white chromosome is going to be on the darker chromosome. The shorter piece of the dark chromosome is going to be on the white chromosome. So how does that relate to the genes that we were just talking about here? So let's do the color coding. So this long one now, if it switches this with this long one, here's the site that crossing over occurred. And so here's that one's original gene. But notice here, because it crossing over didn't occur at the same spot, that it actually switched. And now this darker chromosome has two copies of the gene. It has the copy that it originally had, and it has the copy now that was on the white chromosome. So it's two copies of that gene. And then this one, here's the site, that side of crossing over, because it switched it with this, they swapped. This has no copies of that gene. So I'm going to write that here. So this one has two copies. I'm just going to call it the purple gene because I colored it in purple. And this has no copy of the purple gene. Two copies right here. What is that? That's an example of a duplication. So now this has two copies of that gene. So now what can happen? So now, let's put here, when it has these two copies here, let's write here, if a mutation occurs in one of these genes, a new gene can be, I'm going to put quote unquote born. It's actually not born, but a new gene can occur. So this is the regular gene. Now it has two copies of that, but when the mutation occurs and you have a different order of bases, you can make a different protein, and if that protein is a good protein and does a specific job, then now you have a new gene in this organism, and then if they pass it on to their kids, then their kids have that gene, and so on and so forth, and after thousands of years, it can be a new gene introduced into the species. And so that's an example how gene duplication can lead to, and mutations can lead to new genes. So let me give you an example of this in real life. The human globin genes, an example of that would be hemoglobin. We've talked about hemoglobin with transporting oxygen in your red blood cells. And there are other globin genes as well. So the genes encoding for the various globin proteins evolved from one common ancestral globin gene, which duplicated and diverged and changed. The diverging is caused by mutations. Kind of like these two genes that I was using as an example. So after the duplication events, differences between the genes in the globin family arose from mutations that accumulated over many generations, and the gene copies over many generations. So let's look at an example of this. Okay, so here's your ancestor. Look at here, evolutionary time. So this is back in the day, this is current time, all right? Okay, I'm just going to put long ago over here. So as we said here, that they coded for various, the genes that we now have evolved from one common ancestral globin gene. So that's this, ancestral globin gene. What happened to it? Duplication, just by the same process that we just talked about. So now we look at here, here's a piece of DNA. Now we have two of those genes. That's the duplication part. From here to here, what has happened? The alpha and the beta is representing a mutation. So they've mutated. So both copies individually mutate. So now they're two different genes. And so, so you have mutation, alpha, and I'm just going to use it that they're different genes. I don't have enough colors, I don't think, to show all the different mutations here, but that's the mutation. I'm going to color it in both here. Mutation, both of those. All right, so that's the mutation. Then it says transposition. So let me talk about that. Transposition to different chromosomes. It moves to different chromosomes. Let me explain that real quickly here. If you have, what happens is if this is a pair of chromosomes, let's say chromosome pair number 1, and you have this alpha and the beta gene, so I'll put alpha and beta, these are the mutations. And then here's chromosome pair number 2 that does not have those genes on them. What happens is that these genes can be randomly cut out and inserted into a totally different chromosome that they weren't in there before. So this, what's happening here, the gene is randomly cut out and inserted into another chromosome. This is, we call these sometimes jumping genes. This process is called transposition. So that's this here, transposition. So what they're saying here is these genes are on the same chromosome here. Now, on the same chromosome here, on different. That's why they've separated. So they're on different chromosomes. That's the orange and the blue. And then look at here, then it says further duplications and mutations. So on different chromosomes, again, different mutations. So this one didn't mutate, this one did. So they've duplicated the gene, and then this one mutated, and then these guys mutated again, and so on and so forth. And so that's what we see here over time. And this is the current set of globulin genes that we have. So here we have these guys originated, all of these, we have 7 genes. It says the alpha globulin gene family. So there are 7 genes that we now have on chromosome 16 that originated from mutations and duplications from the alpha form of the original gene. And then on chromosome 11 in humans, we have the beta globin gene family. There are 6 genes we have a chromosome number 11 that originated from the beta mutation, which mutated originally from the ancestral gene. And all of these code for different globin genes. So subsequent duplications of these genes and random mutations, that's what this part right here is showing you here, gave rise to the present globulin genes, which code for oxygen binding proteins. Example, hemoglobin. Oh, this is supposed to be globin. Sorry, guys. So so what they've done is they've looked at these genes and found that they're very similar. The similarity in the amino acid sequences of the various globin proteins support this model of gene duplication and mutation. Since they're so similar, it supports this duplication process and mutation process. So, so what happens? The evolution of genes with novel functions. So the copies of some duplicated genes have diverged so much in evolution that the function of their encoded proteins are now very different. See, these guys here, they've duplicated and mutated, but their functions are still similar. They make proteins that are similar, and in fact, they're oxygen binding proteins. And so those are similar. These guys, sometimes if they've diverged so much that they're totally make a different protein that has a total different function. So this gene duplication creates a situation in which one copy of the gene maintains its original function, while the duplicate may involve a new function. So if this is the original and this is the duplicate, this is the original. The original makes a protein that gives it a certain job, protein A. This one makes protein B. The original duplicate makes the original protein A, makes the new protein B, which gives it a total different function that's totally unique and different than the original. And I put here the example, the anafish gene in fish. I'm going to give you that example in class tomorrow, so I'm going to hold off on that. And so this last little bit talks about this transposable element, which is jumping genes. So this part right here is the jumping genes. Movement of transposable elements, right, which is the jumping genes, or recombination between copies of the same element may generate beneficial new sequence of combinations. Some mechanisms can alter functions of genes or their patterns of expression and regulation. So what does that mean? It says new sequence may generate beneficial new sequence combinations or can alter their functions. So these jumping genes can be good or bad. So let's go back up here. When this gene jumps from one chromosome to another, let's say it inserts it smack dab in the middle of another gene. So now the green gene doesn't work. That could be detrimental, but now you have a new sequence in between here, and so therefore the sequence of A, T, C's, and G's of the DNA with the old sequence and the new sequence may be beneficial and provide a new protein, or it may be detrimental because that gene isn't working anymore. And so therefore these jumping genes can introduce variation to the population by changing the sequence of the A's, T's, C's, and G's, and therefore the proteins that are made. And it can be a good or a bad thing, but over the course of tens of thousands of years, we can get new genes out of it. And so that is where I'm going to end for today. So I hope you enjoy the rest of your snow day, and I'll see you tomorrow.